There we go. All right. Good evening and welcome to the 2025 Peter Doherty Science Awards. My name is Greg de Cruz and I am the coordinator of the PDSA at Ballarat Tech School. I'm also one of the STEM educators here. So I'm going to be your MC for the evening. I apologise for that. And what we're going to do is we're going to go on an adventure. Okay? We're going to celebrate creativity, curiosity, and courage, because those are all three qualities that's required of these people, these special people who are going to tell their stories tonight. So tonight we're going to hear from teams of young scientists who will share their stories of their investigations, the questions they asked, the methods they developed, and the discoveries they made along the way. But before I get ahead of myself, nope, there it is. Hard to get ahead of yourself when you don't have a clicker. There we go. I would like to start by acknowledging the Wadawurrung people.
Sorry, we've got a bit of a feedback loop happening, but our coordinator Albert will be all over that.
That was very cool. Very cool. All right, so let me just start again. I'd like to acknowledge the Wadawurrung people, the traditional custodians of the land and the waters on which we are here tonight. For tens of thousands of years, they've been the land's first scientists, observing, experimenting, and passing down knowledge. Their deep understanding of country came from asking questions and learning from what they discovered. We pay our respects to their elders, past and present, and we're grateful to pursue our own curiosity and discovery on this land. Okay, about the PDSA. The PDSA, or the Peter Doherty Science Award, celebrates more than polished results. They recognise the process of doing science. They recognise the forming of questions, testing of ideas, making sense of unexpected results, communicating findings, and hopefully solving some problems along the way. One thing we love about this competition is how it mirrors real-life research. The scientific method isn't necessarily a recipe you follow step by step, although it can. It's a set of tools that you use in whatever order you need to help you understand what's happening. So this year, entries have come from schools across Ballarat and the wider region. We have teams from Ararat here tonight and Hamilton. It's good to see. Each team will share their work on stage, followed by questions from our judges, who I'll get to in a moment. After all the presentations, the judges will deliberate while you enjoy some refreshments and explore the posters on display out here in the lobby. Okay, so our judges, our esteemed judges. These are very special people in my mind, okay? Because they embody the spirit of curiosity and discovery, which is so important in what we're here to do tonight. So we've got Michael Poulton, Chief Executive Officer of the Committee for Ballarat, a long-standing supporter of the PDSA. Really appreciate seeing you back. We've got Dr. Makoo Kuba. She's a Project Officer at the Walter and Eliza Hall Institute of Medical Research in Melbourne. Makoo has been an ambassador of the PDSA for 4 years now?
Something like that, yeah.
Yeah. Lovely to see Makoo come back. We also have Dr. Sharon Olson, the J.G. King Cancer Research Fellow and Laboratory Manager of the Fiona Elsie Cancer Research Institute. And last of all, but not least, Dr. Jason Kelly. Jason's also from the Fiona Elsie Cancer Research Institute, and these two people have provided so much of their time to give tours to our students, our VCE biology students, and I'm just so grateful for them doing that and being here tonight. Okay, so that's enough from me. It's time to hear from our finalists about their investigations. So please join me in welcoming our first team to the stage. Lucy, Ali, and Olivia, who will be presenting their investigation on how scrolling social media affects our sense of time. So, ladies, let's come to the stage. There's your clicker. That's all right, we'll go back.
Is that going back?
So go back. Yep, go back.
Wait.
Oh, now you're going to see everything in reverse. Now you know the conclusions. Should we?
Hi, my name's Lucy.
And I'm Liv, and this is... Ali, are you coming?
1 second, I just need to finish this reel.
Ah, okay, make it quick.
How long is that reel?
Oh, sorry, it finished ages ago. I'm coming.
This always happens. It's so annoying.
I'm always late to school because of it.
And I never have time to do anything else.
Did you guys know that scrolling can negatively impact your health?
Really?
It can also disrupt your sleep.
And it's customised to keep me engaged. I spend nearly 5 hours on it a day.
Put your hand up if you've ever lost track of time while scrolling. Look around. This is why we wanted to complete this experiment.
Our aim for this experiment was to discover if scrolling on social media can affect your ability to tell the time.
Our hypothesis was that if individuals engage in scrolling on their phones, then they'll lose track of time and go over the 5 minutes.
There are a few variables and safety considerations that we needed to recognise before we started.
Our independent variable was whether subjects were scrolling.
Our dependent variable was how close to 5 minutes the subjects asked us to stop.
And the control variables were the timing method, as well as the instructions given, the age range of the subjects, and the environment the experiment was completed in.
In terms of safety, we needed to watch to make sure that the phone didn't overheat. We did this by not allowing participants to go over 10 minutes.
We also need to avoid too much exposure to blue light. We again avoided this by stopping the subjects at 10 minutes and completing this experiment before 8 o'clock, 1 hour before any of the subjects went to bed.
Finally, we kept everything private to avoid giving away non-consented information. We also made sure that subjects were aware we weren't watching them. We were doing our own thing and waiting for them to tell us when to stop.
Then we began our experiment.
Here's how we did it with Lucy and Liv demonstrating. First, we collected a stopwatch, a sticky note and a laptop and subject 1 with their phone. We then took subject 1 into a room with minimal distractions and placed the sticky note over the top of their phone to cover the time. We then explained the task, which was letting us know when 5 minutes is up while scrolling on either Instagram or TikTok and don't count. Then tell the subject when to begin and start the stopwatch at the same time. Then pause the stopwatch when they say to stop and record the results in a table. Repeat steps 2 to 7 but without scrolling. The subject can sit there, they can fidget, but no more. And then repeat steps 2 to 8 with all 12 subjects.
These are the results we received. Half of the subjects had a longer time when scrolling. When we look at figure 2, we can see that people were further away from 5 minutes when scrolling.
When the median of the raw results is analysed, it is found that people went longer without scrolling. They were also on average 15 seconds away without scrolling compared to 12 while scrolling.
Majority of our results show us that people were further away from 5 minutes when scrolling. We also believe that one of the best methods for finding conclusive results is the difference between each subject's individual scrolling and not scrolling result. The mean of these results gave us positive 8.25, meaning that subjects went 8.25 seconds longer when scrolling compared to not.
Although this experiment was done to the best of our abilities, there's always some things that we can improve.
We need to make sure that the subjects are unaware on what they're being tested on. When they know that we are judging how close to 5 minutes they are more conscious to their time and possibly changing their levels of engagement and therefore their result.
We would also make sure that the experiment is completed on the same day of the week, as people may be less interested in their feeds on a weekend as they've been on them all day, and more interested on a weekday as they're tired and just want to scroll.
So, if we did this experiment again, how would we fix those things?
We would convince the participants that we are testing their memory from what they just watched.
And we would complete it with a longer time frame and more than 12 subjects, as this would give a more reliable result.
When completing this experiment, we needed to remember that everyone's brains work differently. Therefore, everyone would be affected by scrolling differently.
Like how I have 2 border collies at home, recognised as the same dog breed, but entirely different when we bring out the hose. One barks with it and the other plays with it.
If we had to make a conclusion though, we can conclude that the participants lost track of time and were more susceptible to going over 5 minutes while scrolling. Although this wasn't by a lot, this still supports our hypothesis.
A huge thank you to Greg, the tech school team, Miss Yu and Darcy for all their feedback, and our participants for making this experiment possible.
So, we encourage you all to watch your screen time with a watch. Are there any questions?
Okay, thank you, Lucy, Ali, and Olivia. I think that was a very interesting topic. I think it's something that we've all considered from time to time. I like their recognition that people are different. Okay, they went into this investigation probably expecting to see some sort of uniform result, but it turns out, just like Lucy's border collies, different brains are wired differently, okay? And that raises new questions. And I think that that's what science is all about. So thank you. Could you just remain there? We'll pass this over to the judges when you're ready.
I'll go first, Greg. And well done, Team Phoenix. That was fantastic. It's really engaging. You described the problem quite eloquently at the start. You went through how you did the experiment very succinctly. One of my questions that pops into my mind, you said the participants were either looking at TikTok or Instagram. Do you think though the 2 different types of social media may have had an impact? Like if TikTok has a set amount of time for each reel, whereas Instagram might vary, could those have impacted your results?
I think there could be a possibility, but a lot of the time it is very similar on the platforms. Like a lot of the things that are posted to Instagram are actually from TikTok. So a lot of it is very similar, but there may be minor differences. But we don't believe that would affect our results.
And one other quick question. Is this still on?
That's right, you asked the question and I'll...
One other quick question. You used a mean at one stage and a median at another stage. Can you explain why you would use one or another of those statistical methods?
So we used a mean at one point because we didn't see a huge spread in our results, and then we used a median at another point because there was a bigger spread. So when you use a mean for a bigger spread, so when there's a bigger spread, you're better off to use the median because it'll be a better representative. Whereas when there's a smaller spread and maybe one that's further out, the mean won't show it as well because that higher one will bring it up.
Yeah. Charlotte?
Well done. Congratulations on actually being the first group to present, which I know is going to be, really, really scary. So good job on getting through this and also, again, as Jason said, presenting wonderfully and really engaging as well.
Thank you.
This those were really technical questions, so I'm going to ask a very broad one. So now that you've found out a bit more about how scrolling impacts how you experience time, have you changed your habits before school? And have you told other people about it and see what they think?
I would think that I've become more conscious of my time on my phone, and that I'd be able to like think about it more and maybe stop myself from staying on and just going, oh, one more, it won't hurt, because it eventually will.
Yeah, that's fair. Has anyone else got any comments on that?
I have definitely become much more conscious of my screen time. I have definitely been checking my actual phone screen time every day now, and it has gone down quite a lot.
Nice. So something that's really important in science as well is the experiment bit's really fun too, but it also has to have something, an impact. And even if it's just that little thing of, oh, I'm going to be a little bit more wary of my screen time, that's the whole reason why we do this sort of thing. So yeah, I'm glad to hear that it's had some little change in your life. Good job.
Thank you.
Okay, thank you. Thank you. All right, we're off to a good start, I'd say. Okay, so next we have Sienna and Harriet Roberts. They will be presenting their investigation on wind force, angle and turbine energy. So could you please help welcome them to the stage?
Good afternoon, everyone. My name is Harriet and this is Sienna. We're Year 8 students from Marion College, Ararat, and we're here today to talk to you about directional wind force and its relation to turbines. We found interest in this topic as there are multiple wind farms surrounding our area. We soon discovered we had many unanswered questions related to this subject, so we decided to investigate. Our project relates to the real world through the fact turbines produce clean energy, which is becoming increasingly relevant as it does not create any emissions, drastically reducing greenhouse gases that are harmful to our environment.
In our experiment, we used a desk fan in place of a turbine to produce and read wind energy using a simple setup involving our controlled variables, which were a hairdryer, desk fan, ammeter and multimeter. Our dependent variable, which was the angle of the wind force, and our dependent variable measured in milliamps. We also had a larger scale protractor that we used to mark our angles to ensure they were as accurate as possible, and a retort standard boss clip to secure the fan. We used our hairdryer on the highest speed to blow our desk fan from each angle. Our fan was connected to an ammeter and multimeter. Both these devices we used so our data was precise. We repeated this process accordingly, which in our case was 3 times at each angle shown. After 3 trials, we collected our averages in milliamps and graphed this data. This demonstrated that turbines are most effective when the wind force comes from a certain angle, that angle being 90 degrees. The reason for that is just simply because the desk fan or turbine is designed to work like that with the angle of the blade slant.
During this experience, we learned about how wind turbines create electrical energy and store it. This expanded our knowledge on what we already knew and put our minds to the test. We researched different parts of a wind turbine and how they all work together to create energy. This was helpful when designing our experiment. What we discovered was the blades are shaped like aeroplane wings, which create lift, causing them to spin around the rotor. The low-speed shaft connects the rotor to the gearbox. The gearbox increases the slow rotation of the blades into a higher speed rotation that's suitable for the turbines. The high-speed shaft connects the gearbox to the generator, rotating at a much faster speed and transmitting the rotation of the blades. The generator converts the kinetic energy produced by all the spinning into electrical energy. The tower supports all the components and elevates the turbine higher to catch more wind. And the nacelle, which acts as a house for all of the components at the top of the tower.
After the submission of the poster, it was suggested that we dive deeper into the reason behind this 50-degree outlier. After contemplating and researching, we can't find the exact reason why this angle didn't fit our pattern, but we do have a suggestion as to why. The most likely reason that the 50-degree angle was so much higher was because the desk fan was catching the wind on that side due to the slant of the blades, which created lift like an aeroplane wing, causing it to spin faster. Whereas on the other side it was creating drag rather than lift, causing it to spin slower. While this might not be the real reason, it is our closest explanation.
Although our experiment design is an effective way to execute it, our mini turbine still differs greatly from a real wind turbine. Real wind turbines are extremely tall and would catch much more wind force, whilst ours only received what the hairdryer was blowing. Real turbines are also motorised with brakes and gearboxes to control the speed. For example, it shuts off when it starts to spin too fast or speeds up if it isn't spinning fast enough. And given the fact the desk fan is designed to spin with a motor, not wind, we decided making it spin backwards would work for us to act as our turbine. We had many limitations whilst carrying out this experiment, one of those being the fact the hairdryer wasn't secured. If we repeated this experiment, we would secure the hairdryer in a fixed position using clamps and a retort stand to ensure results were consistent. Another limitation being the fact we didn't have a 3-bladed wind turbine, just our basic desk fan.
To conclude, our investigation showed that the optimal angle for wind force on a wind turbine was 90 degrees. We also observed that the reading at 50 degrees did not follow the overall pattern, most likely because the blades on a desk fan are slanted and not designed to catch wind evenly. In the future, Harriet and I would improve the experiment by securing the hairdryer in a fixed position to reduce wobbling, therefore improving the consistency of our results. We may also consider building our own turbine from wood or cardboard to make its blade design and shape more similar to a real wind turbine. Thank you all of our teachers for helping us throughout this project. We couldn't have done it without you. And to Ballarat Tech School, thank you for having us here tonight to present our speech. Thank you.
Well, I don't know about you, but I'm very impressed by that. The level of knowledge, the explanation at the end, and realising that it is an explanation, it might be wrong, it might be right. That's just beautiful. Okay, that's real science. What I really liked too is that I can see that these young people are engaged with their natural environment around them and the technological environment. And they can see how our lives depend on the two of those two things interacting. It's beautiful. That's what we're about at the tech school. So, lovely. We're going to hand it over to the judges for a couple of questions. When you're ready.
Me again. Yeah, well done. Not having much luck with this, so I'll just talk out loud. Yeah, well done, Team Marion College. That was excellent again. You described the setup very well. It was a very ingenious way of setting it up, using a fan and reverse engineering it from there, almost doing the reverse. So that was really well done. And just reading up an outlier, without that 50 degree, it looked like it would have been an almost perfect peak. So that one outlier, science brings up those outliers all the time. Sometimes that's where the discovery is. Sometimes if you can even investigate that to think, oh, well, that's we've tested again as an outlier. That may be ways to refine wind turbines into the future. Have you thought about looking at that into the future? Okay, the tilt of that fan could pick up at 50 degrees. Maybe we could make a more efficient way of doing it. When you suggested that into the future you might be able to make your own fans. Could you base that on those designs?
Well, I think, yeah, we probably could. We tried our best to kind of investigate it as much as we could, but the fan just like slanted. So it was kind of difficult to find the real reason, but yeah.
Yeah, you might be able to get propulsion from multiple directions then. And have you thought about looking on the vertical plane as well? You've gone around the horizontal plane at different degrees from where it is. But I know these wind turbines are on hills, so you get these updrafts and those sorts of things.
In the future, we would definitely consider doing the vertical rather than the horizontal. That would probably give us a lot more to investigate as well. Yeah. So that's great feedback.
Hand over to, I think Michael's got a question.
It's me. That's right. Girls, great job. It's the best explanation I've heard of a wind turbine ever. So your capacity to articulate what a turbine does just shows you've researched your topic really, really well. So well done there. There's a whole lot of physics and mechanics in that you two are born electrical engineers. So well done, and really pleased to hear you say that. I was interested in the outlier piece, and you did mention that was your closest explanation. Did you consider other explanations as to why the 50 degrees became the outlier? What was your thinking around your explanation, given that you chose one as the closest? Was there some other things that kind of entered your mind?
Well, there kind of was, but what we did was we did 3 trials and then kind of found out the average. And all of our trials were significantly higher than all the others. So we weren't thinking it was because of a draft or a wind. We were thinking it was because of the slant. So there was probably other explanations, but that was definitely our main reason.
The important thing there is you considered those other alternatives and you thought about them and investigated them, so well done there. My last quick question is a bit like Moika was saying earlier, is has your views about turbines in your region been shaped in any way based on your science?
Well, driving past the turbines now, I definitely look at it and be like, oh yeah, I know how that works now. So it's definitely, educated me more about those turbines. Yeah.
Well done. Great presentation, girls. Well done. Thank you.
Thank you. I think Jason's angling at a collaboration.
Yeah.
He's very...
I'll get in and advisor on the side.
This is a scientist for you. He's getting very excited about the prospects of the question that your investigation revealed. All right, so our next... Can we answer the next one? Yeah, sorry. Our next team is Evie and Eden, and they have investigated how knowledge of heart rate could promote better sleep. I think that's something that we could probably all do a little bit better of. So very interested to hear what you've found out, girls. So could you please welcome them to the stage?
Good evening, judges and audience members. Which activities could impact heart rate so that we can in turn predict better sleep? Ironically, this idea was born one night when I was lying in bed, unable to sleep. And that got me thinking, what factors impact sleep? Do some activities, if done before bed, influence it? So I jumped at the opportunity to find out. Hi, I'm Eden.
And I'm Evie. Unfortunately, struggling with sleep is a major problem for Australians. According to a report by Professor Robert Adams, around 60% of Australians report at least one sleeping problem occurring on the regular basis of 3 or more times a week. In today's culture, sleeping too much might be seen as lazy, but we never seem to hear about anything about how important it is for us.
Sometimes it feels like it's associated with being cool. Someone walks into class and announces they only got 3 hours of sleep, but you won't hear them doing the same, saying, I've been eating junk food for a week and haven't exercised at all. Sleep is extremely crucial for our physical and mental condition. It is when our brains process the information we've learned throughout the day and when our bodies reset to keep us healthy. Without enough of it, our bodies can't function the way they're supposed to. This is a major reason why we must work towards fixing Australian health. Not just daytime fitness, but the form that often seems to be overlooked.
First, we started by doing some research, and we discovered that having a lower heart rate before bed is directly linked to better sleep. We hypothesized that some activities, whilst potentially creating an elevated heart rate, will lead to a more rapid decline afterwards, resulting in a lower heart rate after a period of time than the initial rate. Our goal was to observe the impact common activities have on heart rate so that we could use that information to predict sleep patterns. Our next step was to select a range of common activities to test to see which could be done or avoided before bed. The activities we tested were exercising by skipping, reading, being on screens by watching a short film, and listening to calming music. We tested this on our Year 10 class and did this by measuring their heart rate after allowing them to rest for 5 minutes to eliminate any interference or any day effects before letting them carry out said activities. We also randomised the different activities and spread them across different days. Also using a de-identifying system for our data in order to prevent any interference. We then measured their heart rate at the set intervals, without allowing them to do anything that may interfere with the data. Each activity went for 7 minutes, except for skipping, which was done in 1 minute to prevent injury.
After doing this, we were able to measure how their heart rate had risen or fell. We found that some activities caused a spike or fall in heart rate, but would be followed by the opposite, supporting our hypothesis. Because of this, we decided to calculate the percentage the average heart rate decreased or increased per minute. Our results showed that skipping caused a decrease in heart rate by 2.6% per minute, music by 0.3%, and reading by 9.7%. Our only activity to increase heart rate was screens, which caused an increase of 23% per minute.
This means that to promote better sleep, with calculated prediction, we can determine that screens before bed should be avoided, and that exercise should be encouraged a while beforehand, with reading done soonest before bed. Some setbacks that we faced during this experiment was occasionally not having enough Fitbits, so then having to measure heart rate manually. We had, however, taught participants how to do this beforehand. And of course, we were not able to measure sleep itself, but rather research to what led to it.
If we were to do it again, we would allocate ourselves more time to explore the subject, as there is so much to be learned. We would experiment on other factors such as eating, different age groups, or even measure sleep itself. I don't know, my heart rate feels pretty high right now. Maybe we should have experimented on public speaking. Perhaps we could even test whether cheap or real dopamine affects sleep differently. After all, screens, being a form of cheap dopamine, resulted in an increased heart rate, but exercise, being real dopamine, resulted in a decreased heart rate.
Overall, this was a fascinating experiment to conduct, and we will definitely go away approaching sleep with what we have discovered. Our hope is that by continuing research, by acquiring knowledge, by spreading awareness, and even breaking down cultural stigmas, together we can all make Australia healthy again, one step at a time. We would also like to acknowledge and thank the Ballarat Tech School for giving us this opportunity, our teachers Dr. Kirsty Kennedy and Mr. Jared Mayo, and our parents for driving us here. Thank you.
All right, well done, Evie and Eden. Again, another fascinating project. I really liked how they recognised that it's such a broad question with so many parts, and they can only, in the hours and time given to them, the scope given to them in the PDSA, they needed to pick one part of that project to attack, and you've done that really well. Very, very good to see. All right, so what do our judges think?
First of all, is this working?
Yeah, just nice and close.
Yep.
Like an ice cream, you're about to buy it.
Okay. Congratulations, really fascinating topic that you've selected and really well presented. One of the things I wanted to ask you about was, what were you watching during the screen time, and could that affect heart rate?
So they were just watching a short film that we found on YouTube. It was like nothing none of our participants had seen before, so it was completely new, and yeah.
So everyone watched the same thing?
Yes.
Yep. And how has, I guess it's been common amongst the questions tonight, how has this affected what you do before you go to sleep now?
Well, usually when I had trouble sleeping, I would try and exercise, but now I know that's not such a great idea to do. So yeah.
Yeah, I agree with that. I often read before bed now because it makes my heart rate lower and calms me down.
Great. You?
Thanks, girls. I loved your hypothesis, and you stated that really clearly right from the get-go, so well done, and you related it to the real world, and I suspect a number of your friends would experience some of those things you mentioned. So well done there. And I liked your analogy about, I only got 2 hours sleep last night. Oh, as if that's a good thing. I'm just a little bit confused with the skipping versus the screen time. In my head, skipping increases heart rate. Don't know much about scrolling. I don't do too much of it before bedtime. So can you just confirm again in the protocol the time between when the activity happened and when you took the readings for me?
So we measured it over 30 minutes. And skipping, it did increase heart rate initially, but then it would drop later on.
So it was a recovery time that was different between them. Okay. So you mentioned Eden. You mentioned, Eden, that you won't do exercise before bed. You won't screen time either, I'm suspecting. Does the time difference between when you do and then when you choose to go to sleep, does that make a difference in the choices that you make about what you might do before bed?
Yeah, so like one of the things we couldn't do was like measure. We didn't have a lot of time to measure, but we do know that exercising should be done like a while beforehand. And the thing to decrease heart rate like right away the most was reading.
And the outcomes of that really to say, hey, look, you're probably not going to choose between exercise or reading, but you may well choose between scrolling and reading. And what we know is that scrolling ain't no good for you if you want a good night's sleep. Well done, girls. Great experiment.
Okay. Well, we're up to our final team for the evening. I'm going to ask Maddie and Winter to come to the stage. And they asked the question most of us have, I imagine, been avoiding, and that is, which has more bacteria, your phone or your toilet? So prepare yourself.
Winter, did you just sneeze and touch your phone?
Yeah, what about it?
Do you know how gross that is? Think about how many germs are on there.
It's not that bad, is it?
Winter, when you come into contact with bacteria and you touch your phone, your device also gets contaminated.
Really? I don't know.
Winter, everybody knows phones are like your third hand. Would you ever go to the toilet and not wash your hands?
Maybe.
That's so gross. What about cooking for other people? Surely you'd wash your hands before then.
Yeah, of course. That's basic hygiene.
Then why does it not carry over for technology? How often would you use your phone?
Daily.
See? You'd not go one day without using your phone.
That's why we run our experiment. We aim to deem which is dirtier based off visible bacteria, your phone or our school toilets. We hypothesised that the phones would carry more bacteria due to their frequent handling and greater exposure to bacteria.
Our dependent variable, or the thing that we observed, was the growth of the bacteria.
The independent variable is the thing that changes. For this case, the objects we swabbed, the toilets and the phones.
Our control variable, or the variable that stays the same, was the areas we swabbed.
And lastly, the extraneous variable, the variable that has a possibility of changing the outcome of the dependent variable. In this case, the incubation times between both trials. To complete our experiment, we needed
16 cotton swabs, 16 petri dishes with nutrient 1000 agar,
8 toilets, 8 phones,
a marker, parafilm,
and a microscope, optional for observing. First, we labelled all of our petri dishes and set aside a cotton swab for each.
We swabbed 8 of our school phone toilets, which are cleaned every day. Next, we actually swabbed the toilets.
We then swabbed the screen and speakers of the 8 phones. After we collected all the petri dishes, we placed them in the incubator at 30 degrees Celsius. We repeated this for a total of 2 trials.
Here we have a photo of trial 1 phones and toilets. When we compare them, we can see more visible bacteria on the toilets than on the phones. When we can see the same pattern for trial 2, as toilets had more visible bacteria than phones.
As you can see from these box plots, both sets of data have a very similar spread, meaning both toilets and phones had similar results, but the toilets had a higher maximum, meaning some toilets had a higher percentage coverage than phones. So what's the answer, Maddie?
Well, this graph shows that across both trials, toilets had more bacteria than phones. The average coverage overall for phones was 46.357%, whereas toilets was 52.1%.
So toilets are more dirty than phones?
Well, not exactly. Bacteria coverage does not equal dirtiness, as some bacteria is beneficial and normal to be on our phones. We need to find pathogenic bacteria for it to be classified as dirty.
So we can't conduct any of the tests we need to do to decipher whether the bacteria is dirty or pathogenic. To do so, we need to carry out biochemical testing and gram staining.
So we decided to categorise by visuals.
We found many variations of yellow colonies, like on samples 1 and 2, but the most common colonies we found were tiny white ones.
We also found large quantities of a white, pink, opaque, large splotch, mainly on trial 1, like on sample 4. We also found a transparent film on about half of our dishes, like on sample 7.
On sample 8, we found a medium-sized colony with irregular edges and a halo in the centre.
We also found dark brown fuzzy splotches like that of 10 and 11, almost like a fungus, but we cannot be sure.
So what did we do well, Maddie?
Well, Winter, we were consistent. We used the same number of samples for each trial and sampled at the same time.
But things that could be improved on include that the incubation times varied between both trials and that the specific objects sampled weren't the same each trial.
If we run another experiment, we would sample the one location multiple times and use a McConkey agar, a selective medium that turns gut bacteria pink or red to help us identify bacteria that suggest their hygiene. In conclusion, our hypothesis was proven wrong because toilets had more visible bacteria than phones.
But Maddie, why does this even matter to me? Do I really need to clean my phone more often?
Although our results came up inconclusive, everybody should be cleaning their phones more often. A study from the RACGP found 11,163 organisms on only 26 phones of your GPs and nurses. 26% of those were found to be antibiotic resistant and virulent, severe and harmful.
Gross. I'm not going another day without a clean phone.
And neither should anyone.
Thank you to everybody for listening, and thank you to everybody who helped us along our journey.
Now, I think you should all go clean your phones.
And I've got just the thing.
Oh wait, I still need the microphone.
Some judge questions?
Okay, well, there's a lot to like about that talk. And I know there are a few microbiologists on the panel that are just dying to ask questions. But the thing that I liked was they were brave enough to reject their own hypothesis. That's real scientific integrity. It's fantastic. As humans, we find it difficult to admit that we're wrong. But it's how scientific knowledge grows, so I think that was wonderful. So I'll hand it over to the judges.
Congratulations, girls. That was a really great presentation. And very topical in many ways. I'm definitely going to clean my phone after this. And, perhaps bribing the judges with gifts may, work in your favour. The question I had was relating to the timing of when you did your swabs. So were the toilets swabbed at the start of the day, close to when they were cleaned, or at the end of the day? And girls versus boys?
So we only ended up being able to swab the female toilets. So next time we would preferably have a male student on our group, so then we could explore more options. But we sampled the toilets on a Friday afternoon between the hours of 1:35 and 3:15 when we had our last 2 science modules for the week.
Great talk, and yeah, definitely close to my heart because bacteriology is my background, so I love everything bacteria. So I find it's really, really interesting and fascinating as well. And yeah, great job on such an engaging way to introduce the topic, and also good use of props too. That's always a winner in my books. I think you mentioned near the end about something being virulent. Would you mind just explaining to the audience what that might mean?
Virulent means like severe and harmful. So in terms of bacteria,
So like the harmful ones?
the harmful bacteria.
Great.
And then it relates to viruses and stuff.
So with
viruses.
Sure. So with that in mind, you did mention things about how not all of them might be bad for you. So do you want to elaborate a little bit more on why that might be, like why there might be ones that are good for us? I think a lot of people forget about that too.
Yeah, so some bacteria is beneficial, and so like there's some beneficial bacteria that live on your skin and keep it healthy, and yeah, stuff like that.
It's like your gut microbes. You've got beneficial bacteria in there, making sure everything's all clean and everything's doing what it's supposed to. It's the same, but some bacteria doesn't belong on our phones. Like with our next experiment, we would be testing for gut bacteria that would most definitely not belong on our phones, and we'd be really concerned about that. Whereas if we found that on the toilets, it wouldn't be shocking, but we'd still be like, our toilets need to be cleaned a bit more. So it depends on what kind of bacteria we're finding and where we're finding it.
Yeah, great response. Thank you.
I thought that was an exceptional response. What did you think, Miku?
That was great. Exactly what I wanted.
Very good. Okay, so that's it. There are our 4 presentations. So our judges have a difficult... is my mic on? Yep. Our judges have a difficult problem to resolve. They're going to be taking some time to do that, and during that time we're going to have some refreshments served out in the lobby here. So remember, those 4 posters are on display. I'm sure these students who've done such a fantastic job of presenting their investigations would love to talk about them. And after the break, we're going to have a guest speaker. So we'll call you back in when it's time to do that, probably about 20 minutes. Thank you. Well, we're now going to hear from someone who is living the journey that many of you may be considering, the path from student to scientist. Okay, we have Tabitha Byron. She's a PhD candidate at Federation University, our own university, and the Murdoch Children's Research Institute in Melbourne. Tab is researching a very interesting topic, and it's close to the PDSA's heart. It is on unconventional T cells in the human immune system. I hope that Tab will explain what I mean by that. Look, I just want to say, as Tab's coming up to the stage, that she's right in the middle of that transition, okay? That really, really difficult period where it's just so messy and so hard, and you have to deal with the kinds of problems that these students have had to deal with, okay? Not everything goes according to plan. You have discoveries, but you have failures, and you learn from them all, okay? So I didn't want to talk you up too highly, but there we go. So Tab's going to take the microphone, and we're going to learn something about her journey. There you go.
Perfect. All right. Can you hear me? Is that on?
Yes?
Yes?
Okay,
cool. All right. So, yes, today I'm going to give you a few insights into what it's like to be a researcher from a PhD candidate's point of view. So for me, it started at Fiona Elsey Cancer Research Institute, actually, in Year 10. So I was posing here with a micropipette that I'd used for the first time, and I was very excited. And then here I am 8 years later now as a second-year PhD candidate. And here I am chopping up some tissue for one of my various experiments. So when I look between these two photos, what's changed? Thoughts on what I was researching have definitely changed, mainly because I didn't know that what I'm researching now existed back then. But there's also been heaps of major learning curves, not just academically, but also how to act in a professional manner and how research actually works.
Sorry, tap that microphone. Can I
Is
just
not
turn that on?
working?
Yeah, no, I
Yes.
think it's just not.
Okay.
Let's just get that one for you.
I was going to say, I'm pretty loud if it's...
Pick up better on the
Okay.
on the feed.
This one. All right. Is that working? Oh, yeah, that's better. But what's stayed the same is my enthusiasm for learning, hence why I haven't stopped studying since high school, and also how much I don't know. So the more that I learn, the more that I realise there's a lot more to learn, which makes every day exciting. So this talk is about science, not geography. But before I start explaining this analogy about maps, I thought I would just highlight that here is Fed Uni's science campus here in Ballarat. All of you guys are regional, you students. So just a heads up that if you're interested in science to study in uni, there are options a little bit closer to home than Melbourne. Okay, so science. We start in years 7 to 10 looking at the really broad, big picture. And then maybe in year 11 and 12, we start to zoom in to one particular area that we like most. By the time you get into uni, you get to decide what you're interested in. And for me, that was human biology or biomed. Once again, at this stage, we're still looking really big picture because we need to get the sort of, broad understanding before we can zoom in. Second year, we start looking a little bit more in depth into a few different areas that sort of are important to our subject. And in third year, we're looking at some really specialist electives that are going to sort of help us decide which pathway we want to take next. For me, third year made me realise that I really enjoy research, so I decided to take an honours year and then continue on to a PhD. And so with our research projects, as you guys would know from your research investigations that you've performed, we've really, really zoomed in on our one particular topic. But we need to continue to zoom out to see that big picture in order to give our research some meaning. So the continuous cycle of research. First of all, we need to kind of decide to do a bit of a research proposal. So that includes identifying exactly what our topic is. We need to figure out what that question is that we're asking so that we can just decide if it's a true knowledge gap. So if somebody's already researched what we're researching, there's no point in us doing it again and reinventing the wheel. We want to be contributing to some novel results. Once we figure out that there is a gap, we then need to figure out how we can answer it. So start looking at the methods and the equipment. And then probably the biggest question of all is the feasibility. So it's all well and good if we've got an awesome question, but if we don't have the funding or the expertise or the equipment to do it, then it's not a question that we can answer. Once we've got a successful proposal, we then kind of go through this loop of planning, collecting some data from experiments, analysing that data, and then interpreting it to figure out what's next. And so perhaps my naive expectation going into research was that you go through this loop a few times and then you end up publishing some lovely, pretty results. But in reality, it looks a little bit more messy. So there's a little bit of backstepping and, going back and forth, hopping around. And then maybe after a while, we're like, yes, we've got some lovely publishable data, let's put it out there. But it doesn't always work out quite right. We have reviewers that will look at our work, and sometimes they'll tell us, nope, you've got to go back to the planning stage. That might sound a little bit tedious, but it's what gives our job rigour. So, we have a responsibility as researchers to be putting out factual information. So this process is so important. All right, so I'm spending 3 years on one very specific question, going through this process pretty much, exploring an unconventional type of immune cell in humans. So just to give you a little broad explanation, our immune cells protect us against infections and cancers on a daily basis, even though you may not be feeling it or seeing it. But there's still a lot that we don't understand about the immune system. And the more that we do understand, the more diseases we can prevent, diagnose, treat, and cure. So if we zoom into our bloodstream, we'll see these sort of red donut shapes. They're our red blood cells, which are doing a very important job of carrying oxygen around your body. But then there's also white blood cells, which are your immune cells. But oh dear, we've got a bacterial infection. So not all immune cells are going to respond to that bacteria in the same way. There's some that are going to digest the bacteria, eat it all up to clear it away. But then they're like, okay, this is a bit too much for me, I need some help. So T cells can come into the rescue, but they can't see the bacteria, so they're going, what's the problem here? And so what the other immune cell has to do is kind of spoon feed the bacteria to the T cells. They have to chop it all up inside their cell and then present it out on the surface. So now the T cell can say, okay, there it is, time to respond. So look at this a little bit like a window. The T cells need a window into other cells to see that something is going wrong. And it's exactly this process that Peter Doherty, the namesake of tonight's awards, found. And that's allowed for so many new and important discoveries in science since then. So T cells take a long time to act. And that's because each individual T cell can only recognise one infection. So this one's going, the one infection I was made to fight. So then what they do is they start cloning themselves to fight off that said infection with an army. They're saying time to clone and conquer. So you might read what I've just told you about in an immunology textbook, but in reality, it was only in the 1960s and 70s that our T cells that we know and love were discovered. And so it's with new equipment and technologies that new discoveries can be made. So in the 1980s and 90s, some new T cells came on the scene. So they were sort of a bit more frequent in tissues. They were seen to recognise infections in a pattern-like way and were perhaps faster acting. So we labelled them unconventional T cells. And so what I've written down the bottom here, new information can bring more context to existing facts. Now that we have these sort of weird unconventional T cells, we've had to label normal T cells, conventional T cells, so that we have that point of differentiation. I'm interested in unconventional T cells, so we're going to ignore normal T cells now. So some unconventional T cells can recognise stress signals. So instead of seeing an infection, they're seeing that a cell is being overworked or stressed. So same thing, there's still a window, but inside they're seeing that the cell is stressed. Same thing, it comes over, I see, time to respond. And so that makes these unconventional T cells incredibly versatile. It means that they can respond not just to one infection, but lots of different infections and cancers, which means that we have great potential to target these cells for disease prevention and treatment. We don't know as much about these unconventional T cells as we do normal T cells. One of those reasons is because we can't use mice to experiment on them because they don't represent human unconventional T cells quite right. So we've had to find some human models, and I'm going to tell you about some of the things that we're using for my PhD project. So first of all, we can get thymus. So you all have one. It's an organ just above your heart that makes your T cells. And what we can do is we can take out the unconventional T cells from the thymus, and then we are able to see how they develop in healthy humans. One of our end goals to this is that we could grow up an unconventional T cell in a Petri dish and then use that to help treat cancer patients. The next thing is that we know a fair bit about unconventional T cells in the blood, but we want to know if they could change the way that they act in different organs. So what we're going to do is get lots of different tissues, and we're going to see if unconventional T cells look and act differently in other tissues and organs. Finally, this is a bit of a crazy one. Healthy volunteers, I will emphasise that, have been infected with strep A on their throat. And so what we've been able to do is get their blood before infection and after infection to see which immune cells are responding. The end goal for this one is to find some immune targets that we could develop a strep A vaccination for. All right, so to conclude, research projects were really drilled in on one particular topic, but we need to zoom out to see biology to give it meaning. So make sure that you're focusing in your science classes. So we're sort of going through this loop-de-loop with the research cycle, but it's all very important to give our research as much sort of truth or accuracy as possible. And so at the moment, I'm going through this process looking at a very important T cell in your body because the more we understand about them, the more diseases we can prevent, diagnose, treat and cure. So the last thing, which I noticed pretty much all of you guys did in your presentations, is as scientists, we like to acknowledge everyone who's helped us along the way. So I'm very lucky to be part of two lab groups, one here at Federation University and also down at MCRI. So thank you to both of them. For the sample sources as well, it wouldn't be possible to do human-based research without getting samples in. And a special thanks to Greg and Miku for giving me the opportunity to speak today. I might have waffled, so if there isn't time for questions, that's fine. I'll be hanging around afterwards as well. Thank you.
I think there are time for questions if anyone does have any. Probably everyone feels a little out of their depth.
Yeah.
Look, I just want to make a few comments. Oh, sorry, Michael.
Can I ask one?
Yeah, go for it.
At what point did you decide? Fantastic and great communication of your science.
Thank you.
Really well done there. And I don't know science very well, but I've got a bit more idea about T cells now. But I'm just curious as to at what point in your academic journey did you decide hey, this little thing like this, I'm going to focus on for the next 3 years?
It's a good question. I didn't do anything like this in my first project. So in honours, just for those who don't know, honours is like a 1-year program. So it's kind of just like a little mini PhD that gives you like a bit of a taster of research. So you can sort of tack that on to the end of your bachelor degree. I did that in something to do with immunology, but completely different. And I decided I like the immune system. I kind of want to stay in this space. It's probably a bit of a sad but true answer. This was the project that was available to me. So I have a fantastic supervisor here in Ballarat who's an expert on unconventional T cells, and he offered me a project in this space. Of course, I sort of knew what I wanted and didn't want, so I was able to kind of, figure out that I wanted to accept this project. And the more I've learnt about it, the more excited I am. So, yeah. Yes.
Tamitha, congratulations on that presentation.
Thank you.
I think that's your enthusiasm for science is fantastic and mirrors, I'm sure, many of us that are scientists in the room. I'm really keen to know when you were doing, and you described beautifully the transition from high school through to undergrad and then eyes open where to from there. When you were thinking about in third year, what am I going to do before choosing to go into straight science and honours and then into sort of more research, what was sort of grabbing, what other things may have grabbed you back then?
Yeah, okay. So for me, I chose to do biomedical science because I wasn't sure if I wanted to be a clinician or if I wanted to be a scientist. So I was kind of thought that was a great way to sort of get into learning the knowledge that would kind of be required for either way to go.
Sorry, Dab, could I just, what's a clinician?
Yes, like a doctor. So so sort of the person that you go and see if you're sick, pretty much. That was what I was looking at doing, kind of thing. So, good point. I was sort of tossing up, bench side or lab side, pretty much. And then by the time I was in maybe second year, I was like, okay, I like the lab more than I like people, I think.
The thing we're meant to say quietly.
I think I decided that I would be better suited to the lab. I'm a very emotional person, so I don't think that I would have done well with sort of desensitizing myself to treating patients and that sort of thing, but I still wanted to make an impact to, making people healthy and stuff like that. So I knew that if I stuck with biomed research, then I had that opportunity to potentially give back in that way, so it would still be rewarding in that light. Yeah.
Fantastic.
Yeah.
So, why did you choose this topic?
Yeah, so this topic was sort of it was already kind of my supervisors were looking in this kind of space. And so it was kind of the next logical step, which sounds a little bit, crazy. But yeah, pretty much we'd already had my kind of lab group as a whole had already looked at unconventional T cells a lot. So we had a few next steps that would kind of get us closer to figuring out how they could be beneficial in some immunotherapies and that sort of thing. So, yeah.
Now that you're going to become an expert in unconventional next step that you can see on the horizon?
So more translatable outcomes, I suppose. So at the moment we're looking at very sort of fundamental research, so just trying to figure out how these cells work in a healthy individual. There is already some work that's being done in specific disease contexts like infections and cancers. But yeah, I would like to get closer to those kind of research projects at some point because they're going to be closer to actually helping people get better from serious diseases. Yeah.
It's a much broader question, but did you ever think you were going to be a scientist when you were young?
No, I actually thought that I was going to be an artist. So that was my first one. Yeah. But I think that honestly, there's a fair bit of creativity involved in science.
things like this. I like making my slides pretty, and we have to make posters as well sometimes, so making our posters look nice with graphics and stuff. And there's also creativity in terms of how we spend our time. So there's not many jobs where you can kind of walk in and to a certain extent decide how you're going to spend your day. I can choose when I want to be in the lab. I have a bit of flexibility over, okay, I've got an appointment tonight. I'm not going to start a 10-hour experiment in the lab. I'll leave that for tomorrow. Not every person's experience is the exact same in science, but for me that's something that I like, that there's a bit of flexibility involved as well. Thank you.
Is there time for one more?
Go on. Everyone's,
On the spot.
this is fantastic. Go on.
So, for context, the reason why Tab has an acknowledgement from me as well is because we both do martial arts together. That's how I know Tab. I didn't realise until after I met her that she's also a science person, and I was like,
pretty cool. Small world.
So, with that in mind, how important do you think it is to maintain, I don't want to say hobbies, but activities that are outside of science?
Yeah, it's so important. So yeah, for me, I've been doing karate since I was 12. So that's been something that I tried to make sure I had that as my sort of thing that I would not ever stop doing. Even when things got tough during sort of year 11 and 12 and started to get to exam season, I kind of wanted to still be there at least once a week because it was a bit of an outlet. So yeah, highly encourage you all to continue with your sport or your creative outlet or whatever, even when things get a bit busy and stressful, because it's so important. Yeah.
Okay. Well.
Cool. Yeah.
No, I think that was, yeah. Round of applause, please. Thank you. Wow. When questions come from all sections of the audience, you know that things are going well. It's lovely to see. Look, a few things sprung to mind when I was listening to Tab's talk. First of all, knowledge is power. So learning more about these unconventional T cells might one day help us improve treating cancer. I think 1 in 3 people, is it? Or is it, sorry, the cancer experts over here. How many people, 1 or 2 or 3 people will get cancer?
I think it's about 1 in 3.
Yeah. Okay, so it's job security. Yeah. I also thought there was a refreshing amount of authenticity. I think Tab really told a story about her life and, what drives her. And I think it was very relatable, which is nice because, as I said before when we were introducing Tab, that she is like one of you people in the future. You can relate with her. It was lovely. And I also liked the messiness. The messiness. In school, we can't help but stick to beautiful textbook versions of the scientific method, and that's sometimes true, but there's a lot of forwards and backwards, and there's a lot of revision that happens in science. And if I understood this correctly, the T cells that Peter Doherty won the Nobel Prize for not discovering, but discovering the mechanism around, they are fewer in numbers compared with the unconventional T cells.
Can be, yeah.
Yeah?
There's still probably more of them,
Yeah?
the normal classical ones.
Okay.
Sort of thing. But yeah, the unconventionals still have their place. They're probably about 10% of your T cells.
Right, okay. So, I mean, is it a misnomer to call them conventional or unconventional T cells?
Probably.
All right, well, that's history for you.
Yeah.
Okay. So, look, before we move on with the PDSA awards, I just want to make an announcement. We would like to recognise the achievements of all of the finalists and people who submitted posters that didn't get through to. Am I getting told to? Okay, and teams that didn't get through to the finals. Crest. Sorry, let me just change the slide so you know what I'm talking about. Oh, that's Tabitha. Okay, CREST stands for Creativity in Research, Engineering, Science and Technology. So these are awards from the CSIRO, that's Australia's National Science Agency, and they celebrate student-led scientific inquiry. So the criteria for these CREST awards are almost identical to the PDSA. What they're based on is students defining their own questions, conducting experiments, analysing findings and using teacher guidance. They're the unsung heroes. Teachers in the room, put your hands up. Yes, there we go. Come on, there's another couple of you. I know there is. There's one back here. Okay, you've done such a good job supporting your students get through to this stage. Okay, so all of our finalists tonight will be receiving a Bronze CREST Award. And some of the non-finalists have also been selected for a Bronze CREST Award. And if you want to know what that consists of, we've got it up here on the screen. So they will be handed out to the finalists after the presentation. Okay, so enough talking from me. Now's the moment we've all been waiting for the Peter Doherty Science Awards. So before I announce the winners, I want to acknowledge the organisations that made this possible. Collins Booksellers and West Lab have generously provided prizes, and the Committee for Ballarat have generously funded a lot of the cash prizes. So yeah, they've just been an amazing partner. All right, so I'm going to invite the judges to the stage now. Each judge is going to present a different award, and I've asked Miku if she could provide the judges' reflections before we go on to talk about the awards, announce the awards.
Mike, I can project my voice as well.
You can try, Mike.
All right, so I've got cue cards because I can't remember everything that I want to say because otherwise I'll waffle. So I've been a part of this award for, again, a bit over 4 years maybe, quite a while now. I always forget. But it's always a real privilege to be invited back and participate in this each year. Greg's already mentioned this numerous times, but it's always really incredible to see young people going out of their way to give it a go, because so many people don't. They just sort of go, oh, there's no way I'd get it, and won't even make an attempt to try. So the fact that you guys actually tried, let alone, got to this stage, that is highly commendable. So definitely congratulate yourselves, because most people will never, ever consider giving it a go. So that's already a huge thing, especially at your age. It's also really wonderful to see family and friends supporting them throughout this whole thing as well. It makes such a big difference to have that support, no matter what you do really, but for this as well. So thank you for showing up this evening. It makes a big difference. So trying to keep this succinct to the point as much as possible. Highlights. Everyone showed a thorough and depth of knowledge in their topic, and they really highlighted why it mattered to them. Everyone made an attempt to say, like, this impacts me in some way. And and I'm sure everyone here agrees, this is how science starts, and it's really, really important that you relate it back to how it impacts you or people around you. You had good use of scientific language, but you also, and I'm sure everyone else noticed too, we noticed efforts for you to try and make it a little bit more relatable to everyone else. So we always talk about making sure that it's understandable to a lay audience. So as scientists and science communicators, we also have to make sure when we're explaining what we're doing, we want to make sure that everyone can understand what we're talking about, because if they can't understand what we're doing, there's no point in doing it, because it just stays with you, and that's not how things progress. Knowledge has to be passed on. So making sure that you're taking the time to go, oh, this means this, and blah, blah, blah. It's really important, and it's a skill to be able to do that. So good on you for doing that throughout. We also noticed, so Jason and I popped in a little bit earlier, a couple of weeks ago, and looked at the posters, and left, along with the tech school staff, a lot of feedback. And it was really lovely to see that this feedback had been taken on board. It showed that you actually, took initiative and tried to make it better. That's, again, what Tav said. We always try, as scientists, to make things better, to improve, and just hone in on that and make and take in what other people have said. So good on you for doing that. Great use of comedy. Again, science is all about communication and engaging your audience. So, importantly, telling the story. It's always about a story. You've got this idea and you want to tell people what you did because it's exciting. So thank you for going out of your way to make it entertaining for all of us to watch your talk. I know it's not easy at all. I'm terrible at that kind of stuff, so I really enjoyed that personally too. But to summarise, you all demonstrated skills that real professional scientists need, as you saw from Tab's talk as well. Stay curious, ask questions, and always wonder why some things are the way they are. And we're very excited to see where this experience takes you in the future. Who knows, you might end up like Tab or whatever else you do. I'm sure it'll be amazing. So thank you.
All right, thank you, Maku, for those kind words, and I think they were quite honest. All right, so we're going to have Dr. Jason Kelly, present the Communication Award. Okay, so Adrian's going to pass those certificates over. Let me just explain what the Communication Award is for. This award recognises teams that effectively communicated the scientific thinking, clearly explained their methods, and engaged both scientists and general audiences. Okay, I'll hand it over to you.
Thank you, Greg, and it gives me a great deal of pleasure to present this award. As Miku said, and Greg has said, communication is very important. What Tab didn't say is next week she's off to Perth to present a poster at a conference there. I'm also off to Perth to present a poster where there are more than 200 posters getting presented at this conference. It's a very important way to present your data. So learning these skills at your age is a wonderful, wonderful achievement. And so the winner of the Communication Award, everyone did a terrific job, but I think the judges were unanimous with this. The Communication Award went to Maddie and Winter from Phoenix for their toilet versus telephone. So congratulations.
Do you want to say anything?
Do you want to say anything? Thank Greg as the teacher.
It's the Communication Award. They communicated fine. All right, so next we'll have Dr. Sharon Olsen present the Highly Commended Award. So this award recognises the outstanding experimental design, rigorous methodology, and thoughtful interpretation of results.
I've
you.
gone blank on names.
It's about the names on the sheet.
It's on the sheet. Winning. Well, first of all, I'd just like to congratulate all the speakers tonight. I think you've done an amazing job. The opportunity to participate in this type of award really gives you skills that will be translatable throughout the rest of your life. The Highly Commended Award goes to Sienna and Harriet. Congratulations.
There you go. Whoa. Trophies. You've heard her? Yep. Well done. Okay. It's the business end. So Michael Polton will present the overall winner. The 2025 Peter Doherty Science Award goes to a team that demonstrated exceptional scientific inquiry from question to conclusion, combining strong methodology, honest engagement with results, and clear communication.
Greg, I'm going to let them just sweat for a moment or two.
Oh, ouch.
I'm the only non-doctor on the stage, so I'm going to depart. But Community for Ballarat has been long associated with these awards, and we are delighted to provide the cash prizes for them. Why? Because we actually think that a region like ours, a city like ours, is far better when you've got young scientists with curious minds delivering for our city into the future and to our regions into the future the innovation that we need to move forward as a community. So that capacity to keep asking the question as to why, to keep challenging, to keep being able to communicate what it is you do, to be able to counter misinformation with scientific rigour is really, really important. So we are delighted to be a part of this. We're delighted to partner with the tech school. FECRI, it's a world-class scientific institute just over there. And if you can say that about scientific institutes, you're pretty lucky. So the fact that we've got amazing researchers in this city is a part of the reason that we do as well as we do. So thank you to FECRI and to Miku. It's been, I think, Miku, sorry, I should know that by now, shouldn't I? Certainly been a number of years we've been involved, and it's fantastic to have you back here today. So the winner, as declared by the judges today for their experiment on scrolling, is Lucy, Ali and Olivia from Phoenix College. Well done, girls. And there's an envelope for each of you.
Thank you.
It's really heavy photo, don't
Thank you so much. Thank you.
Get your photo. Okay. There we go. Okay, so congratulations to all our winners. I'm sure it wasn't an easy decision. I'm biased. I'm very biased. But I honestly think that these 4 investigations, these presentations that were done this year, are probably the best that I've seen in the PDSA. So, you know, take that for what you will. Can the finalists please hang around after the event? We'd like to take photos of each of you with your teachers and the judges, if that's possible. And we also have a few other things to give out, some gift vouchers and things like that. So, yeah, if the photo is not worth it, then hopefully the gift voucher is. All right, so, look, I just want to say before... Oh, sorry, I'm being told that...
for each of the students and the schools next to their posters.
There you go. So you can put it on up in your wall. You can take down whatever it is, whatever. I don't want to even guess at what's on your wall, but you know, you replace it with your poster, your scientific poster, and you can think about toilets and phones. So, I just want to finish off by saying congratulations to everyone who took part in this year's competition. It does take courage. It takes courage at all stages. Yes, getting up and talking to people is difficult, but it also takes courage to listen to feedback. I think sometimes it takes the most courage to listen to feedback and to acknowledge that results didn't go the way that you had planned. I've seen lots of that tonight, so I'm very, very impressed. A huge thank you to our sponsors, the Committee for Ballarat, Collins Booksellers and Westlab, as well as the CSIRO Crest Awards for recognising scientific inquiry in our region. And thank you to the teachers, mentors, and of course the families. Okay, you've played such an important role in embedding this curiosity in these people tonight. Lovely to see. Look, I'm just going to finish off with something a little esoteric. I think of science as humbling us continuously. Every single experiment just humbles you. You learn, I think Tab was saying it, you learn how much you don't know, okay? And that's a fantastic feeling after a while. Yeah? No?
I know nothing.
Learn to like it. Learn to like the pain. But it's the way that we learn. It is the way that we learn. And what I thought was really remarkable is the questions that you were asking, that's the start of learning something. If you don't have a question in your mind, conscious or unconscious, it doesn't matter what you see, it's not going to make sense. You're not going to take it on, okay? So as the judges said, don't stop questioning. Questions, okay? They're great. So the best scientists have gratitude for the mistakes they make because That's where the real discoveries hide. Okay, so most people think science is about certainty. I'm always saying that it's not. I would say it's quite the contrary. Science is about being comfortable with uncertainty and remembering that all the models are wrong, but some of them are useful. What matters is asking good questions, as I keep saying, and being honest about what your answers tell you. So with that, I'd like to say thank you very much for attending tonight's event. I hope you've enjoyed it as much as I have. And I look forward to seeing some of you next year.